Hello, this is Yingjie Zhang. I'm currently an assistant professor in the Materials Science and Engineering department at the University of Illinois. I received the Beckman Young Investigator Award in 2021. Here I'd like to share with you my scientific journey in the past 4 years. So Dr. Arnold Beckman's legacy has been truly inspirational. He was actually a UIUC alumnus, obtaining bachelor's and master's degree here. He was one of the best inventors of scientific instruments, famous for inventing the pH meters, spectrophotometers, and many, many more. He is also one of the greatest philanthropists of all time. The Arnold and Mabel Beckman Foundation has been supporting young scientists for a few decades, and I'm very grateful for the support I have received in the past 4 years. The Beckman supported research has been focused on molecular species in liquid, especially the small molecules that are usually less than 2 nanometers in size. Examples are water and metabolites in living cells, organic solvents, anions, and lithium solution complexes in batteries, and reactants, intermediates, and products in catalytic reactions. These molecules are crucial for the chemical, electrochemical, and biological functions. However, the existing microscopes in the community are pretty much only sensitive to solids or macromolecules, and small molecules are mostly hidden in the existing tools, including optical, electron, and scanning probe microscopes. To bridge this gap, we have developed a method to image the interfacial liquids. The method is called electrochemical 3D atomic force microscopy. We achieved 3D imaging with atomic scale resolution by measuring the tiny force between the interfacial liquid and the sharp tip. Using this advanced tool, we have made many exciting scientific discoveries. At flat substrates, we found that the liquid density exhibited damped oscillation behaviors. And in heterogeneous substrates, there are rich emergent behaviors that depend on the local morphology and curvature of the substrate. We also observed that the interfacial liquid structure strongly modulates the overall electrochemical energy and conversion and storage functions. So while this is all nice, there is a limit in this AFM tool, and that is I can only image within a few nanometers from a solid surface. And this is not enough for many biological, electrochemical, and other systems. So, to overcome this scaling gap, we have designed a new type of microscope. Instead of doing the force mapping using a mechanical probe, we've used a fiber probe, which enables the transmission of mid-infrared light. And this enables us to measure the mid-IR transmittance as a function of wavelengths and the 3D spatial coordinates. From this four-dimensional data set, we will be able to determine the chemical composition and distribution of liquid species in the microns and larger scales. And here is initial setup for this endoscopic FTIR method that we have already built. Currently, we have achieved IR transmission through liquid over almost the full mid-IR spectral range, and we have achieved micron scale resolution. And right now, we are working towards sub-micron and eventually nanometer scale resolution. So as a way to verify how this developed instrument prototype works, we have measured pure water and found that the results quantitatively agree with the standard infrared absorption properties of water. So this means that we have been able to very accurately determine the local chemical composition and concentration of liquid species. And besides the continued instrument developments, we're currently using this endoscopic FTIR setup to monitor the internal processes of living cells, battery cells, and catalytic reactions. With that, I'd like to thank my group members and collaborators. And once again, I really appreciate the support from the Arnold and Mabel Beckman Foundation. Their support really supporting my bold, ambitious ideas and help my lab develop the instruments that I showed here. So that's all I have. Thank you for your attention.